Joining us today in this edition of Research Connection is Dr. Cristina Lanzas. Thanks for joining us.
Thanks for having me.
So tell us a little bit about yourself.
Okay, so I'm associate professor here in infectious disease. I'm originally from Barcelona, Spain, where I did my veterinary degree. From there, I did a master's, PhD, and a postdoc at Cornell University in upstairs New York. Both my master's and my PhD actually were focused on applications of mathematical modeling to animal nutrition. My postdoc was on infectious disease epidemiology. So from there, I have kept in the area mostly of infectious disease epidemiology. So I was for a few years assistant professor at the University of Tennessee in Knoxville, and then I moved here, which I have been here for 4 years now.
Very nice. And tell us about your research.
So our lab really are focused in the areas of infectious disease epidemiology and modeling. So broadly what we do is we are what we call a dry lab. So we try to combine data with computational and mathematical models of transmission, and our goal broadly is to understand mechanisms behind transmission. So what are the main drivers of a transmission of a pathogen, how we can actually use these models also to evaluate interventions, because oftentimes when you are trying to evaluate interventions for infectious disease, basically you don't have a chance to do any experimental work. And so we sort of do what we call in silico experiments with the models to try to evaluate scenarios where we evaluate interventions. And so for the last 10 years, my focus has been mostly on enteric pathogens as well as an antimicrobial resistance, but we have a broad number of collaborations looking other things like such as vector-borne disease. So it's pretty broad, anything that has to do with pathogens. Just to give you a few examples of the types of projects that we work, for example, for the last 10 years I have been working a lot with a pathogen that is called Clostridioides difficile, which is actually mostly a human pathogen. It's also an animal pathogen that you acquire in healthcare settings, so it's mostly a hospital-acquired pathogen. And so for over the years, we have been developing models to try to understand basic aspects of the transmission of these pathogens. We have used the models to test interventions, to evaluate the role, for example, of environmental transmission. And lately, we've been focusing in aiming to understand vaccination. So as vaccines are being developed and they are currently in clinical trials, our goal is using computational approaches to try to understand basic questions about how we deploy vaccines. So another area that we have been working very actively also is on antimicrobial resistance. So one particular interest that we have is trying to understand how, for example, we can use surveillance data that gets collected through different agencies such as FDA, CDC, and how we can actually repurpose that data to evaluate and answer questions about control strategies. So oftentimes that data, the main goal why the data is collected is just to describe what are the trends on antimicrobial resistance. Another area that we've been working is just broadly understanding transmission of enteric pathogens. So for the last 3 years we have been working on a NIH-funded project where we are working with a collaboration with Kansas State University, where we are carrying on transmission experiments with cattle, where basically what we do is we have a relative large group of animals, about 70 animals. We do transmission experiments, meaning that we take a few of those animals, we inoculate the strain that we can actually follow, and we mix those animals with a bunch of susceptible animals, and then every week we try to assess who is getting infected. On top of that, we use real-time location systems. So these are systems that provide information about the location of the animals in a very high resolution. We are talking about a second here. And so we use that data to infer sort of the contact structure of that population. And so we use that to try to answer basic questions about transmission of pathogens, such as, for enteric pathogen, how much of transmission is due because of direct contact between the animals, how much of that also is due to social interaction of the animals versus environmental transmission.
Interesting.
So.
And do you always work with cattle or?
So most of my work, both since I had done PhD, has been in cattle, but right now we are really more focused about developing methods. So we are very open. So we have a project with swine too. I do a lot on the human side too. So I work a lot with hospital-acquired infections in humans. So basically, we are very, when it comes to the host pathogens, we are very broad in that sense. At the end of the day, our main goal is try to, push the envelope of the methods that are being developed to characterize transmission.
Thanks for joining us.
You're welcome.
And best of luck on everything.
Thank you.